I'm Martin Beck, and I worked at the Structural and Computational Biology unit from 2010 till 20.
And you were a group leader here at the EMBL?
Yes, I was a group leader at the EMBL.
Was this your first role as a group leader?
Yes, I came out straight out of my postdoc. I was 32 years old and naive when I started, yeah.
And I'm sure you're no longer naive, Martin.
I guess yes, and that's a very important aspect of being a group leader at EMBL. I came as someone who was technologically motivated. I was very good with technology, but I didn't know what hypothesis-driven research was. They still hired me. And 6-7 years later, maybe, I had become the scientist that operated in the way I do today. And it's being at EMBL is like putting you next to a very strong magnetic field that is driven by very rigorous and excellent science in molecular biology. And at some point you just flip and you know how to do it. And that's exactly what happened to me. That's
a great metaphor. So let's start at the beginning. How did you get interested in science, and what was your background in science before you arrived at EMBL?
My parents are both scientists. They are both in chemistry, and they had one major advice when I left the house, which is I shouldn't study chemistry. My plan at the time was to become a professional musician, but to make my sponsors happy, I studied biochemistry. And at some point I realized I had a good talent for biochemistry more than for music. And then it happened, I finished my diploma, applied for a job at Rockefeller before my PhD. They took me. I learned some cell biology, and then I had done two, as I said, I was very technologically driven, two educational phases, a PhD in Martin's Reed, mostly focused on cryo-electron tomography, and a postdoc at the ETH in Zurich that was mostly focused on proteomics. And after that I came and literally combined these two technologies in my laboratory. That was my starting point.
And what drove your interest in technology?
I was always interested in going to the bottom of things, understanding something very deeply. I was always a good coder. I can program a little bit, and that gave me an edge to deal with very complex scientific instruments and get good data and to process them. And that was my early career, yeah.
EMBL, of course, has a rich history, and indeed potentially was founded with the concept of developing new instrumentation to open up new areas of biology. Was that also part of the motivation for you?
Not initially. Actually, the cryo-EM equipment that EMBL had when I was hired was not as good as what I was used to. And then I knew years later that that's not what makes EMBL, that you have plenty of money to always have the newest instrument. They really buy that for you if you desperately need it, but then it's possible. What was interesting is that I essentially experienced what they call in cryo-EM the resolution revolution at EMBL. And there was this time of detector envy where other institutes started to have direct electron detectors, and we still didn't. But it was fine. They let us use their instruments, and we got good data. And then there was this one moment in time, I remember this very well. It was a faculty retreat in Stromberg, where we got Ian convinced to fund...
Ian Matai, the
Ian
director.
Matai,
yes.
Yeah. We got him convinced to fund a very fancy cryo-electron microscopy facility with all the newest instruments. And that was the start of cryo-EM service at EMBL that then took off and become a very important part of also the service operation.
And you, of course, were already aware of the power or opportunities with cryo-EM from your PhD studies
yes,
with Wolfgang Baumeister.
Very much. But
so you came with a prepared mind.
I came with a prepared mind, yeah. At the time it was a niche field with not many people being active. Now it's a huge field. But it's interesting to know that EMBL from the very beginning, from the times of Jacques Dubochet, has always been part of the development, and they had some very, very important contributions to that.
Yeah, it's been very interesting chatting with both Jacques and also Alistair McDowell and hearing about their eureka moments when they discovered how to vitrify samples and deal with holding the water in samples, which I guess underpins the subsequent field.
Yeah, this is. I have a small anecdote there. I don't know which year that was, but it was before the Nobel Prize for JAG. Ian actually had asked me to write like a couple of pages assessment of his work because he wanted to nominate him for some prize. I don't know which one. But at the time I went in all the old papers and also into the EMBL archive to look at JAG's old reports. And what was very, very impressive is, so he essentially had developed this technology to vitrify water by plunge freezing against the belief that this would be possible. There were literally papers out there saying this cannot be done. And then he had that, and in this very moment, he had a very illustrious crowd of visitors. There was all kinds of famous people who came to visit Tokayashu, or people from the LMB, and so it was the who is who of the later cryo-EM field who actually came to visit JAG and to learn about what he did at EMBL. And that shows what is really the strength of that system, that it's outgoing and that it's ready to spread the newest discoveries.
Yeah, I think that story really illustrates some of the wonderful things about EMBL, giving people the freedom to work on important problems, even when they don't know how they're going to solve them, and giving them some time and space to do things rather than forcing you to know the answer and the method before you get a grant. But also when you do make a breakthrough, feeling that it is your responsibility to share it with the field and train people.
Yes. There is another aspect to it, though, that I would like to highlight, at least an aspect that was important to me. To be a scientist at EMBL means to do science among friends. And for me, that's the essence of EMBL spirit. It means you can run into any door at any time and impose your scientific problem onto people, and they will be ready to help you with their know-how and their technology. And for the very moment, they put their personal career development and their personal goals at the back, and they will be there to help you. And that is impressive. And I didn't know that it's not necessarily a given once you obtain an excellent position elsewhere. But it is not so obvious once you left EMBL that this is the treatment that you're going to get.
Yeah. It's by no means universal, and I'm delighted to hear you say it. It was also my experience and the foundation of my own scientific career. And it's been enlightening for me in conducting these alumni chats to hear the same sentiment expressed by so many successful and famous scientists, that this was so important for them. I think it's something that EMBL should be proud of and cherish for the future. And for those of us who have trained at EMBL and then left, I think it's also important for us to take that philosophy with us when we train people. And that, I think, actually, and that's underappreciated as my personal belief, but that's actually EMBL's biggest contribution, that the spirit goes out there and the alumni take it and they try to foster it at home. Tony made this point today about the MPI CBG in Dresden, which is one beautiful example for this, but there are so many. You find it everywhere on the planet, and that is something which is absolutely priceless and a huge contribution to science. Who are the people, would you say, in your career, before or during your time at EMBL, who most influenced you or helped you?
I had three mentors before EMBL who were all important. Günther Blobel for showing me the big world after I had left little East Germany. And then Wolfgang Baumeister and Rudi Ebersold were my mentors, who, from both of them, they are actually very different, and from both of them I learned a lot. At EMBL, there were so many. I had a lot of fun with my two heads of units, Per Borg and Christoph Müller, and both of them are very good heads of units in the sense that you can always bump into the door and ask for advice, and they're genuinely interested in your success, which is great. And then in terms of doing science among friends, I also, in the preparation of the talk that I have to give, but I cannot put all of this information in, I checked with whom I co-published at EMBL and whom are the people that I think I mostly interacted with and where my interactions were most important. And the two lists are actually not really the same. There's some people with whom I published a lot of papers, and some I think the interactions were very important. So I want to mention Oshulya Barabas and Edward Lemke, who were the two closest friends whom I owe a lot of things, a lot of advice, also a lot of private interactions that were very important. There was the KIM crowd of the time, which was John Briggs and Carsten Sachse. Those were important to me. Later on, Julia Mahamid had joined the team. And I also interacted a lot with one person with whom I never published a paper, and that is Jan Korbel from Genome Biology. We just happened to be a mental match in completely different fields, but we could meet and discuss and help each other with getting opinions on the present problem. So also that was a very delightful interaction.
Yeah, and I think that really is an important point that speaks to the spirit of EMBL you mentioned. It isn't just about, we do an experiment together, we all get paper out of it. People share very generously thoughts and ideas and can stimulate and inspire you, even if you don't necessarily end up co-publishing.
Exactly, this is very true. And I want to give you another example for that, which is also in EMBL story. So I told you already that I'm a structural biologist, and when I came I was mostly interested in structures. And I once gave a unit seminar that was attended by Bernhard Hampholz, who was a postdoc with Anne Frossi. And he came to my office afterwards and said, I would like to talk to you about some strange observation. And he showed me a confocal movie of a developing Drosophila embryo, where you had some patch of nuclear pores inserting in the nuclear envelope. That was a very surprising observation, but it was at the same time rock solid in his data. And we had a 2-hour conversation. Afterwards, I hired him, and I started to collaborate with Anne Frossi on Drosophila development, although I knew literally nothing about Drosophila biology and developmental biology. And we ended up publishing a number of papers, with Bernhard being the driving force. And I guess Anne and myself and also Yannick Schwab, who had joined the team, had a lot of fun with this, where we had discovered a maternal nuclear pore complex biogenesis pathway that only operates in mothers, and then that nuclear pores are inherited to the early embryo, where they facilitate rapid nuclear divisions. And that happened to me as a structural biologist. And today, I have a Drosophila lab in my department because of that experience, and that shows what is possible.
And absolutely, it's a great story and another good example of one of the strengths of EMBL, bringing people together with very different complementary expertise and working at the interface between different subjects or areas. And working at that interface is something you're now famous for also. Perhaps you could tell us a little briefly about some of the biological work that most excited you when you were here at EMBL. I mean, you're famous for nuclear pore analyses.
Yes. So my entry point was, as I said, it was today. I think it's a simply defined question, though a huge technical challenge. Nuclear pore complexes are complicated. They have a very complicated architecture. A human nuclear pore consists of 1,000 proteins. And you can imagine this as a very intricate three-dimensional puzzle where your pieces of the puzzle are simply proteins, molecules that you need to fiddle together. And I had imagined this as my end career goal, and I wanted to get started with this at EMBL. And when I started, there were models that tried to attempt to address this question out there, several, but they would contradict each other. And that was my entry point. I was like, look, there's these 3-4 different models, they all say something else, so obviously that's an important question. It was that simple. And then at EMBL, you could really combine different technologies to look at this problem. And one smart decision that we made at the time is to focus on generating information that facilitates data integration. So we could essentially capitalize on a lot of published data that were available, and we were generating data that allowed us to click the pieces together. And that was a very smart strategy that actually allowed us to, at some point, suddenly be at the forefront at this field, although we were a junior group. And that it was something very interesting that happened to us. So that was the puzzle of the nuclear pore. And then I already told you that while I've been at EMBL, I kind of became a hypothesis-driven person by beforehand. I really had training in structures and systems biology, so I was on the opposite end of the scale. I would go systematically about things. And then I became interested in nuclear pore complex biogenesis, and we had the story with Anne Fossey that I already mentioned. We also became interested in how HIV enters the nucleus, which is a very intriguing question still. Many pathogens need to deliver a message into the nucleus, either to manipulate the host or to infect the host. And HIV does that as well. It needs to deliver the retrotranscribed genome to the nucleus, and for that it has to interact with the nuclear pore. And that has been one of the mechanistic questions that we focused on in the last couple of years. Actually, still in collaboration with Hans-Georg Kreusli here from Heidelberg. So it's really a project we started quite some time ago in Heidelberg, and we are still doing it, and it has turned out very rewarding.
You made reference to being involved at this expansion of the resolution revolution in cryo-EM, and also to the importance of data. I think these two things are obviously highly connected. When Jacques and Alistair developed the vitrification methods, perhaps the computational power wasn't there to take full advantage of everything you could do in the cryo-EM, and that's been something that's emerged. How do you see that developing in the future, that relationship between computational power? Everyone talks about AI and machine learning, and methodology and instrumentation.
I see it both as opportunity, but also with a bit of nervosity, and I will try to explain why. It is obvious in my field, and I think it will become obvious, or is already obvious in many other fields as well, that whoever is in control of outstanding computational power, whoever can pay for the electricity bill, may dominate a field. And this is now great for being at the Max Planck Institute, which obviously has very good infrastructure and has the means to pay for the electricity bill. I think it's great for EMBL, who will be able to do this as well. I see it with a bit of nervosity for all these smaller institutes, all the universities. Will they be able to keep up with training artificial intelligence? Because this is computationally so expensive, but those tools are obviously very powerful. And the second reason why I see it with a bit of nervosity is because the industry has the most computational power. And I realized that in the newest version of AlphaFold, my team told me that there's this web server, the source code is still not available, but in this web server you cannot run viral proteins, right? Why is that? So AlphaFold can now do small molecules, so you can do predictions that are potentially relevant for drug discovery and drug development, and they sit on the code and they don't publish it. But also whoever has a lot of computational power will be able to make more calculations, and that is something we all have to get used to, that beyond generating data, having the computational power to look deep into them is the next edge, I'm afraid.
I share your nervousness about the future, also with respect to European science and its ability to play in this field. I actually see some parallels with exactly what you're describing, with the situation that almost happened with DNA sequencing and genome sequencing, because this was, as it became industrialized, and of course EMBL played a major role with Willem Ansorger and his colleagues in making instrumentation that could actually sequence at scale DNA in the first place. But there was then this move with Craig Venter and the Tiger Corporation, and there was a risk that genome sequences would, and their relevance for drug discovery and drug targets would be completely owned by the pharma commercial world. And when you look now at the tremendous underpinning to so many fields of discovery from anthropology to microbiomes that are made possible by having that information freely available. I think we all owe a huge debt of gratitude to key people like John Sulston and others who fought so hard to break that monopoly on the knowledge that would come out. And it worries me a little bit that as helpful as AlphaFold has been, if the future iterations are owned and restricted by a single corporate entity, that actually then it becomes a block to development.
Yes.
So I wonder how we can, I mean, how are you going to deal with that problem?
So first of all, we have to see how severe it becomes, but there may be some kind of pay for access model that, you know, it's imaginable if the next amazing version is there of AlphaFold, you simply pay for using it. But that's not yet even the worst case scenario. The worst case scenario is we don't get access to this code anymore, and it's held in a box, and it's actually used only for industry. That is my nightmare. So what can we do about it? It's just code.
Yeah.
I think it can be copied. And
or replaced with an improved version.
Exactly, replaced with an improved version. So we have to be aware of these bottlenecks. And maybe it's even good that industry now competes with basic science, and we have examples where we need to catch up, yes. But also there are some of the smartest brains on the planet involved. And if that becomes a bottleneck, I think automatically scientists will counter-steer. I think that the intellectual problem is not the biggest problem. The problem is that you need the money to afford computational power.
Yes.
So you need science funding to be sustainable enough that we can actually compete with industry. That will become very, very important. And it's a challenge. It is a challenge.
It is absolutely, and it's important to raise awareness of that and also make it clear to the people who fund science so they realize that. And personally, I think EMBL may be an excellent opportunity to fund this deliberately in a transnational organization that's not simply meeting the needs of one particular small group, either of industry or a single nation, but who could perhaps provide
that is
tools.
One thing. Another thing they're strong with and where they could engage and make a difference is they are politically within Europe very well connected. They are very good with talking to politicians in the different countries. And that is a point that they could make and take into politics, because there we need to make a change to make sure we're not overrun.
So you quite correctly have highlighted some risks, potential problems. We haven't given up hope yet. There's absolutely hope that we'll still find a way through. There's also, of course, fantastic opportunities.
Yes.
And something that always impresses me in hearing you talk, Martin, about your research, is you just see this vision of like two scales of biological analysis with light and then electron microscopy and then atomic resolution crystallography, and somehow it starts to feel like these are going to meet together. Yes. And that must be one of the most exciting things possible.
I couldn't agree more. You essentially put, nowadays when I do grant writing and I illustrate my vision, this is more or less what I write.
Feel free to put that in and just send 10%.
Fine, I'll do that, Angus. No, I literally think these two disciplines are currently merging. I think many people have realized that, but I also do think, and now this is dreaming up, is I think we will have atomically resolved models in cell biology. There will be atomic resolution structures of subcellular segments. Okay, in the next decade, they're not going to be yet complete. There will be some problems with sampling the exact lipid composition or knowing the exact post-translational modifications. But we're going to have them, and we're going to run them in the computer. We're going to see what happens. And I think such computer simulations of parts of cells will become predictive, and they will be the next hypothesis generators that will allow us to motivate new experiments. That is very clearly the vision for both of these fields, and I think it's happening right now.
Yeah, and that's a really exciting vision, and I'm sure you're going to be one of the leaders taking that forward. When you left EMBL, as I know very well you have gone on to great things, leading a Max Planck Institute in Frankfurt. As you're aware, we're here celebrating the 50th anniversary year of the EMBL organization. Perhaps as we conclude our discussion, you could just say a few words about what EMBL has meant to you in your life and career.
I'm going to say a sentence that I think many alumni say. Thus far, it's been the best time of my life. My family enjoyed the time in Heidelberg so much. I enjoyed my training. EMBL has made the scientist that I am today. And it was amazing. I stayed maybe a little bit too long. And the reason I say that is I went through a phase where I saw my best friends departing. And maybe it would have been better if I didn't have that experience, but it was okay. So I figured that also I had aged a little bit, and then I moved on. But I'm very, very thankful for that experience. I'm very thankful for all the means that I've been given as a very young group leader. And obviously this has worked out for me in an amazing way. And sometimes I would like to, push back and do it again, but unfortunately that's not possible.